Greetings, and thank you for the opportunity to talk to you guys today. My name is Christine Foreman. I am Professor of Chemical and Biological Engineering and the Associate Dean of Engineering at Montana State University in Bozeman, Montana. My research group studies life in icy environments. Today, I'm going to talk to you about microbes in the cryosphere. This photo was taken around midnight in southwest Greenland, and it shows icebergs calving from Jakobshavn Glacier, the fastest moving glacier in the world. I need to give a shout out to my colleague and friend, Dr. Jill McCookie, for sharing some of her slides with me for this talk. Thanks, Jill. So, as microbiologists, why should we care about the cryosphere? Interestingly, from a biological perspective, our Earth is a cold planet. Most of the Earth's surface is covered by oceans, where temperatures are below 5 degrees Celsius, and 80% of the terrestrial biosphere is permanently frozen. Although the origin of life and the emergence of microorganisms on our planet remain unknown, some proposals for a cold origin of life have been made, like Snowball Earth. The term cryosphere was first introduced by Dobrowolski in 1923 and is derived from the Greek word cryos, meaning cold or ice, and refers to areas where water can be found in its frozen state. So what I'm going to do right now is show you a little video clip from Antarctica to hopefully get you into the mood of thinking about these cryospheric systems. Here we go.
Okay, this is one. Okay. Two.
Hello!
We get out there by 5, looking not quite alive, but the whole needs to be chipped again. Why do you take so long? Take so long. Did you have a nice walk? We did. You can tell it's cold in here, okay? And I'll do this tape. Are you alright, Chris?
Yeah.
Are you going to film man? Winch?
Yeah. Yes, he went.
Okay, stop.
He went.
You ready to go drop it?
So Chris, what? Yeah, slow down, slow down.
While it seems extreme, we know that life survives and in some cases even thrives in ice. The image in the background here is of a backlit snow pit in West Antarctica, showing the layering of snow bands, where summers appear as lighter bands because of more snow, and winter layers are darker and more compressed, with less snow and more wind. This layered snow gets compacted, trapping microbes as well as evidence of the atmosphere, as can be seen in this inset image from our cryostage microscope, where you can see individual ice crystals abutting up to one another. And in the liquid, in the interstitial veins and triple junctions that form between those crystals, you can see bacteria present. So what happens is, as the liquid freezes and you form these ice crystals, the bacteria get concentrated into these veins and triple junctions along with other nutrients. So the cryosphere includes environments where water appears in its frozen form. It includes permafrost, glaciers, ice sheets, and sea ice. I will go over each of these briefly today and then talk about some of the special adaptations that allow organisms to survive in the cryosphere. When we think about our cryosphere, it's experiencing a lot of changes, right? So it's currently more affected by global change than almost any other regions on our planet. Glaciers and ice sheet melting contribute significantly to sea level rise. Sea ice thickness and sea ice extent is declining rapidly. Permafrost regions are warming, and the extent of seasonal frozen ground is decreasing. Snow cover is declining with these crazy dynamic seasonal shifts. Lakes and rivers are freezing up later, and ice is breaking up earlier than in the spring. This past summer was the warmest summer on record for the Northern Hemisphere. So we're going to start off by looking at different areas of the cryosphere that can be seen in this expanded map of our planet. So we'll start first with permafrost, which is shown here in the purple regions of the map. Permafrost is soil, rock, or sediment that remains below 0 degrees Celsius for 2 or more consecutive years. Permafrost underlies most of the Arctic land area. In fact, parts of Siberia, it's up to 1,500 meters thick. This impermeable layer of ice below the surface shapes the landscape. It prevents water from draining away in summer, leading to super high water levels and wet conditions in many areas. Approximately 24% of the Northern Hemisphere's ground, or 19 million kilometers squared, can be found in permafrost. These areas are also important repositories of organic carbon. When they melt, they release this carbon and other greenhouse gases to the environment. And here I want to highlight two nice permafrost reviews of the permafrost microbiology, Jensen and Tass and Gilchensky et al. So in this cartoon, we can see that permafrost is overlain by a seasonally thawed active layer of soil, shown here in brown, over the gray permafrost. In the lowland areas, the ground is divided into polygons that are separated by ice wedges. As the active layer deepens and thawing water pools, thermokarst lakes develop at the surface. At high elevations, permafrost thaw results in drainage of the soil moisture. As you move down from the surface to the permafrost below, the amount of water and carbon decreases, as does the amount of oxygen and nutrients in the soil, setting up redox gradients that microbes can exploit. The arrows in this cartoon indicate gas exchange, as permafrost thawing releases methane and increased microbial activity of respiration, photosynthesis, denitrification, methanogenesis, methanotrophy, and methylotrophy all exist as this environment warms. There's a diversity of microbial life present in permafrost, with dominant bacterial lineages being proteobacteria, actinobacteria, firmicutes, and bacteroidetes. Fungi are the primary eukaryotes, and methanogens and methanotropes round out the archaeal diversity. Here in this image to the right, you can see a nice ice wedge coming in from the active layer down into the permafrost, and then this thermokarst lake where you have ebullition of the gas coming, so you have methane coming up and bubbling up to the surfaces here. The next area of the cryosphere to explore is snow, shown here on the map in green. Microbes are transported in the atmosphere and get deposited with snow. The snow matrix is generally characterized by low temperatures, high ultraviolet radiation flux, and scarce nutrients. This leads to relatively low abundances of microbes in the snow. A few years ago, we examined the role of microbes in nucleating ice and found them to be widely distributed in snowfall. This ability to grow ice crystals at warmer temperatures using ice nucleation active bacteria has been exploited by the ski industry to make artificial snow using a strain of bacteria called Snowmax. And you can see here the inset on the right showing the production of snow. And what they found is that when you use Snowmax in producing snow, you can get about a 90% increase in the amount of snow that's created for the same amount of energy there. So really helps to create more snow if they use these ice nucleating bacteria. Sea ice and ice shelves are shown here on the map in white and blue, and they cover approximately 25 million kilometers squared of our planet. They're also an important habitat for the diversity of microscopic and macroscopic life. Sea ice forms, grows, and melts in our world's oceans. It waxes and wanes with the seasons, as can be shown here in the maps of Antarctica, where we have large changes in the cover, sea ice extent cover based upon the time of year. So shown here in September, the Antarctic winter, you have a maximum, and in February, at the end of the summer, you have a minimum of the extent of sea ice surrounding the Antarctic continent. This sea ice also has a large impact on albedo, as it can be seen in this image on the bottom left. So when sunlight hits the white ice, it gets reflected back into space. But when it hits the dark oceans, because that sea ice is no longer there, the radiation gets absorbed, leading to more warming and a positive feedback loop that then tends to melt more and more of that ice. Sea ice is interesting, and there's an entire microbial cycle occurring with the initial colonization happening during sea ice formation. So what happens is you have these ice crystals slowly start to rise, this kind of fragile ice. The microbes attach to these ice crystals. They get wafted up and advected onto the bottom of the sea ice. And then a diversity of microbes get sort of caught and entrained in this sea ice. So then what happens is you have winter, right? And so microbial activity is low, it's cold. And then you get to the period of time where spring starts again, and we start to get more solar radiation coming. So as solar radiation increases, there becomes this giant bloom of sea ice algae. And this bloom of organisms happens and biomass starts to accumulate. The bloom only ends either when nutrients run out or when the sea ice melts back in the late spring and early summer, releasing these organisms back into the water column. Glaciers and ice caps are shown here in black, and the ice sheets with black dots. And these serve really as a museum or an archive back into our past. They can offer high temporal resolution records of temperature, precipitation, gases, pollutants, microbes, going back hundreds of thousands of years. So as you can see in the inset cartoon on the right, what falls in snow gets trapped and eventually gets compressed, and eventually trapped in the ice. So as it gets deeper and deeper, it gets more and more compressed, and then you eventually get to this glacial ice where you've sealed off the bubbles. And those air bubbles become sealed off and contain evidence of past atmospheres. In 1997, Michael Bender, Todd Sowers, and Ed Brook published a paper in the Proceedings of the National Academy of Science describing how we can use these greenhouse gases, such as CO2 and methane, trapped in glacial ice, as a proxy for past climatic conditions. Which to me brings up sort of an interesting question. So we know that microbes exist in snow and ice. What role, if any, do they have in this gas record? Now, the glacier ice sheet ecosystem can be divided into three sort of broad realms, as are shown here in the cartoon to the right. The superglacial, on the top surface of a glacier, the end glacial environment, which is the interior of the glacier, and the subglacial, or the areas below. Of the three, the end glacial environment is the least studied. So we'll start, and I think this cartoon showing mountain glacier ecology, hydrology, and geomicrobiology is a nice sort of overview capturing what's going on, because it shows these three glacier regions. So you start at the top with the glacier surface, where sunlight fuels carbon fixation by algae and cyanobacteria. This in turn is then used by heterotrophic bacteria. Dark organic matter gets deposited on the surface of the glacier. This reduces the glacial albedo and begins melting in to form structures known as criconite holes. Within these holes, microbes are attached to particles, forming a biofilm and creating a hotspot for microbial activity. As the temperature warms, cracks form, and the surface melts, nutrients and cells start making their way down through these crevasses and moulins to the glacier interior and also to the subglacial zones where chemolitho autotrophs fix inorganic carbon. Glacial meltwater coming off of these systems feeds organic matter, cells, and nutrients to downstream ecosystems as well. And I've highlighted a few nice review articles on glacier microbiology on the left here if you want more information. We're still continuing talking about superglacial environments, and this cartoon from our work in 2016 with Dr. Foster shows the tight microbial coupling that occurs within a criconite hole, which leads to these really high levels of microbial activity. The extracellular polysaccharides produced by the microbes helps to aggregate the dark particles together and again reduce that glacial albedo, like I showed earlier with the sea ice. Interestingly, these microbes from superglacial environments are often highly pigmented, as can be seen on the Petri plate shown here from one of my former students, Marcus Dieser. Also, you can see this high level of pigmentation on glacial surfaces in the images from a paper by Alex Anicio and colleagues, where they show the impacts of pigmented microbes on the Greenland ice sheet. Here we're looking at the end glacial environment. So after snow falls on the world's ice sheets, it eventually becomes compacted into this porous layer called firn. At a depth of about 60 to 100 meters, depending on the site, the firn transforms into this solid ice, permeated with just trapped air bubbles. So ice cores, such as those shown here, are cylindrical samples drilled from glaciers, and they've played a critical role in our understanding of the past climate of our planet. These cores may reach several kilometers in depth and contain a record of that atmosphere from modern pollutants all the way back to ancient pollen, bacteria, and gases. This core shown here in the middle was taken from the West Antarctic ice sheet and shows a really nice, easily distinguishable layer of volcanic ash. You can see other things that get trapped within the ice as well. As I mentioned, ice cores preserve microbes along with the more typically measured chemical components. A few years ago, we examined ice cores from birds stationed in Antarctica from about 21,000 years before present, at a time during the last glacial maximum. And we compared those to ice cores from the West Antarctic ice sheet divide from about 15,000 years before present during the last deglaciation period. In comparing these, we found differences in organic matter composition and microbial diversity based on the environmental sources and deposition patterns onto the Antarctic ice sheet during these distinct climate periods. When you examine these microbes, they're often quite small from being trapped in cold, nutrient-poor environments over time. And the image on the bottom left here shows ultramicrobacteria from some of Mateva and Brunchley's work. And so you can see these cells, and the scale bar is 0.5 microns. There's a diversity of subglacial environments out there as well, including hypersaline lakes recently found in Devon Island, Canada, as well as lakes below volcanic craters in Iceland. And there's hundreds of lakes below the ice sheet in Antarctica. There are also basal-rich debris layers that you can see forming these nice kind of ribbons along the bottom surface at the base of glaciers. These systems share some commonalities, including being chemosynthetic and supported by chemical energy rather than photosynthesis. All of these studied to date have actually been able to show, recover microbes from them. And so this comparison that came from Dr. McCookie, who's at the University of Tennessee in Knoxville, is a nice comparison between Blood Falls, which is a subglacial feature, and subglacial Lake Willans. And so again, they share certain commonalities by being chemosynthetic, chemical energy driving that energy cycle, but their difference is in terms of the organisms that are present and the environment itself. You can see nicely on this sliding scale at the bottom. So subglacial Lake Willans was warmer than Blood Falls. Blood Falls is saltier. There's higher diversity in subglacial Lake Willans, but Blood Falls may appear to be a more stable community over time. As I mentioned, microbial cells exist in all of these subglacial samples collected to date. On the left, you can see this striking image of Blood Falls in Antarctica with an inset of microbes cultured in the McCookie lab. In Blood Falls, iron metabolizing bacteria liberate soluble iron, which oxidizes at the surface to result in this beautiful red kind of spew coming out. And to the right is a cartoon explaining the environmental conditions and resident microbes found in Blood Falls, which is essentially an external conduit of subglacial water below the Taylor Glacier in Antarctica. When the conditions are right, this water spills out of the terminus of the glacier and enters into Lake Bonny. And so you can see this beautiful red stain coming out of the glacier at the terminus and then spreading on to Lake Bonny. These microbes persist and actively cycle iron, sulfur, and carbon. So here's a couple of articles about subglacial environments from Jill's work. When we look at the cryosphere, we might first think that these environments are lacking in life, but this is far from the case. The cryosphere microbiota have an unexpectedly large genetic potential, with taxonomic as well as functional diversity present. The variety of microbial niches and associated processes are nicely summarized in a review article in Nature Reviews Microbiology by Boschus et al. In 2015. The cartoon shows how microbes employ diverse strategies to gain energy. For example, in the top panel, snow and superglacial ice are dominated by photosynthetic microorganisms, including cyanobacteria, brightly colored ice and snow algae, and heterotrophic bacteria. Chemolithoautotrophic communities, however, dominate in the subglacial environments, where microbes use the minerals and carbon substrates in the bedrock to generate energy through redox reactions. The bottom panel, B, shows life in snow and ice. Heterotrophic bacteria inhabit all dimensions of snow and sea ice, including the thin first-year ice, thick multi-year ice, melt ponds, and ice structures called frost flowers. Cyanobacteria can be found in snow and surface melt ponds. sea ice algae form large aggregates at the bottom of these melt ponds. These algae, and especially diatoms, also inhibit the brine channels in the bottom surface of this ice. Shown here is a really nice compilation of metadata from a paper by Macario et al. In 2015 in Research in Microbiology. This article highlights the range of abundance and community composition of microbes in different snow and ice areas of the cryosphere. The pie chart at the top left shows when and where the data came from, whether it was Arctic, Antarctic, or alpine environments, and then also the sampling season, whether it summer, spring, or winter, fall. The abundance of cell counts per mil of melted snow and ice are shown in the box plots, with the whiskers giving the highs and the lows. Community composition at the phylum level is shown in the heat map at the bottom. And to the right is another figure from this paper showing a cartoon of the food web in snow and ice. Those green dashed arrows represent interactions present in sea ice, while blue continuous arrows represent interactions in glacial ice, and red dotted arrows are those in snow. So how is it that microbes survive in these cold, often inhospitable environments? Studies have shown that these organisms are not endemic. Rather, they can be found globally and are really characterized by their unique adaptations to life in ice. These organisms use strategies at all different levels of cellular physiology to retain metabolic and enzyme activity at really low temperatures. Characteristics of cold adaptation include the expression of cold shock proteins, creation of extracellular polysaccharides or EPS, the presence of compatible solutes such as glycine, betaine, glycerol, trehalose, etc., which help to reduce the freezing point of the cytoplasm in the cell, scavenge free radicals, and help stabilize cellular membranes. There's also structural adjustments of enzymes, maintenance of membrane fluidity through the use of unsaturated fatty acids and desaturases, the production of antifreeze and ice-binding proteins. Microbes also regulate metabolic traits related to energy generation, compounds and mechanisms involved in stress resistance, oxidative stress resistance, and cold adaptation, as well as characteristics of that cellular envelope. We can use these unique capabilities to bioengineer better solutions. For example, in my lab, we're studying biosurfactants, or green detergents, which help organisms separate complex substrates they feed on into easily to metabolize droplets. These green detergents have several advantages over their chemical dispersants. They're safe to release into the environment, they work at lower concentrations, and at sub-freezing temperatures. They can also be used in bioremediation efforts to stabilize biodiesel products, and even allow us to save some energy and wash our clothes at lower temperatures. We're also studying microbially produced pigments as natural sunscreens. For example, a student in my lab was looking at this really beautiful violacean pigment, this purple pigment, which is isolated from an organism in Antarctica. This pigment has been shown to possess antioxidative properties, and it has a potential as a natural sunscreen. So at this point, I want to wrap up. I hope I've convinced you that there's lots of life in the cryosphere, lots of really interesting places in which to study that. All of this work, I can't help but thank all of my students, colleagues, all within there. And at this point, I will stop, say thank you, stay safe out there, and ask if there's any questions.